Hello and welcome back. Today we'll be discussing the second part of the biotechnology chapter, that is biotechnology 2, the technology and its applications. Okay, so what is biotechnology? We have to get some product or service on a commercial scale. That is the main purpose of development or progresses we make in the field of biotechnology. Okay, and the three critical research areas in biotechnology include how best we can make that catalyst to perform in its optimum, how we can improve that biocatalyst, that organism, which is usually a genetically modified microbe or any other cell, so that we will get the purest form of our product or get the service in the purest form. Two, we are all creating optimal conditions so that they will be working at their maximum efficiency. So creating optimal condition through engineering for catalyst to act, then only that catalyst is a genetically modified organism, so that we will be getting that product at the purest form easily and cheaply. Three, separating the product from the bioreactor, that is called downstreaming process, and to get that heterogeneous protein or that compound. That is our ultimate aim, to get that compound, which may be an enzyme or a hormone or a therapeutic, at the best form, at the cheapest way. So we have to improve the downstreaming process. Okay. And we want to apply our findings, our progress as we made in the field of biotechnology, mainly in agrochemical based agriculture. So agriculture is number one because our population is ever increasing. So to feed that extra mouths, we are having large number of mouths to be fed. So that is prime concern. How to improve our agriculture and how we can improve agriculture. One is by chemical based agriculture. Two, organic agriculture. And three, genetically engineered crop based agriculture. Of course, we know that during Green Revolution, we could improve our agriculture production by three times. But why we could improve that? Mainly because of improved varieties. We use large number of improved varieties of crop. That is one reason only. How the new improved variety was produced? By hybridization. But mainly due to the use of better management practices. Three, because of use of agrochemicals. So we have seen that we use a lot of pesticides and fertilizers, which are very, very costly. So farmers cannot afford the cost of production. They will be incurring losses. So we cannot further depend upon chemical based agriculture. So fertilizers and pesticides will also pollute our environment. Ultimately, they will be boomeranging. We ourselves, we are all damaging our own body, our own health, and our own environment. So we have recognized this. Okay, so we cannot depend on agrochemicals for increasing agriculture, and we can only have a genetically modified organisms used. What are genetically modified organisms? Where these organisms are synthesized or produced by gene manipulation. Genetically, you are all bringing deliberately changes, and such genetically manipulated organisms are called GMOs. And why we are all producing GMOs? What are the advantages of having newly formed plants, animals, fungi, bacteria, etc., whose genes are modified? Why we are all modifying the genes? We are having certain aims. These GMOs will be having more tolerance to abiotic factors. So even if there is more drought or more flood or salinity in the water in which they are all grown, no problem. They are all tolerant. No damage will be occurring. They will be reducing the use of pesticides and fertilizers so that we are all not using any pesticide or fertilizer. So reduce the reliance, dependence on chemical pesticides. Why? Because plant has become themselves resistant to the pest. We are all making themselves resistant to pest. So pest resistant crops. Then reduce the damage, loss after harvest. These products cannot be stored for long time, and so they will be easily perishing and destroyed. So they have to be marketed and used immediately. They are having less shelf life, so they will be subjected to post harvest damages. How you can reduce that damage so that they will be having long shelf life? Then increased efficiency of mineral usage. The plants are made in such a way that they will be judiciously utilizing the nutrients. They will take only very small amount of nutrient for the soil and will maximum utilize these nutrients. So that as they are all absorbing small amount of nutrient from the soil, soil fertility is not lost. As soil fertility is not easily lost, we need not have to add fertilizers. Okay, so we are all becoming free from the use of fertilizers. And enhance nutritional value. How you can improve the quality of the product. And one classical example for improving the quality of the product is by the invention of new variety of rice called golden rice. See, children who are all growing in countries where the staple food is rice. Rice is very rich in carbohydrate, but very poor in amino acids and vitamins. These children who are having the rice as their staple food are not getting sufficient amount of vitamin A, which is highly essential for our sight. So these children are very often having deficiency of vitamin A, that is causing eye problems. So how can we solve? We can add vitamin A to the rice. Thus we genetically produced yellow colored rice, that is golden rice. So when the children are eating this golden rice, not only they are getting starch, but also getting vitamin A. So enriched and fortified food products we are all producing so that all the nutrients, minerals are available to all people all over the world. Okay, now we are all having a number of examples where we are making plants resistant to pests. The classical example is the BT varieties of plants. We have successfully produced BT varieties in corn, rice, tomato, potato, soya bean, etc. What is this BT? Bacillus thuringiensis is a soil growing bacterium which is producing some insoluble crystals during their sporulation period. These crystals are insoluble. Why they are all producing? They are producing themselves as a mechanism for protection. Okay. These are all insoluble crystals. That is why they are all in the form of crystals. But we studied these crystals and found that when they are all solubilized, when they are all converted a solution in alkaline condition, they become toxic. And the toxicity is sufficient to kill insects. So they can be used as an insecticide. These are all proteins which are all under the control of a gene, which we call as cry gene. So we are having the cry gene present in this BT variety, which is responsible for cry protein. This is responsible for the cry protein. CRY, capital CRY, cry protein. There are so many different types of cry protein which are all effective against different types of insect groups, like Lepidoptera, Coleoptera, Diphtheria, etc. So we studied them and found that there are so many different types of cry proteins which are all effective against a particular type of pest that is attacking a particular type of crop. We identified there are different types of cry proteins which are all under the control of different types of cry genes. We isolated that cry gene from this bacteria and gave it to the plants so that the plants are themselves capable of producing the cry crystals in their body parts. When that insect is coming and eating on, feeding on these tissues, these insects are pests. This cry protein is entering into their intestine, and under the alkaline condition of the intestine, in the intestine, that is the mesenteron or midgut of the insect, the alkaline condition will make the protoxin. Cry protein, crystal protein is a protoxin, is converted into a soluble toxin. This toxin is soluble. Now, this toxin will make small pores on the epithelial cells of the intestine through which water enters, that will lead to the bursting of the epithelial cells. So, intestine will burst and the insect will die. So, any insect that is coming and eating on these plant parts will die. So, no insect will come and eat it. Thus, we have made pest-resistant variety of plants. Clear? We are having a number of such crops where we are having cry 1 AC, cry 2 AB, controlled cotton bollworm. Cotton bollworm is a notorious pest that is attacking the flowers as they open in cotton, and that will be causing severe damage both in quantity as well as quality of the cotton. So farmers were suffering a lot because of the damage caused by the attack of this bollworm. And now cotton plants are made resistant to the bollworms by introducing cry 1 AC and cry 2 AB. Similarly, cry 1 AB that control corn borer in corn, CMAs. Okay. These worms are coming and eating on them, but now we have made that corn plants resistant to the attack of corn borer by introducing cry 1 AB gene into the plants. Okay. So, only thing is that we have to understand which is the target pest, which is the target plant. That is why we say that specific, the crop and targeted pest. That is the basic. The choice of gene depends upon both the crop, whether it is cotton, whether it is corn, like that, and also what type of pest against which you want to have resistance. Accordingly, we have to select that particular cry gene and incorporate into that one. Okay, that is called cry gene. Now, we have also used biotechnology for making plants resistant against the attack of parasites. See, tobacco plants, they were all attacked by some organisms which we didn't know for long years, which caused some characteristic swelling on the roots of these plants, and they will be having the characteristic swellings, and this disease is called root knot disease. Root knot disease of tobacco. When plants are suffering from the root knot, there will be no nicotine content in the leaves, and there is no quality, and the entire crop is a failure. Because there is no quality for the tobacco leaf, quantity of the nicotine determine the quality of the product. Okay, so here the leaves are very, very low in nicotine content, and farmers knew that these plants are having some swellings on the root, so they call it as root knot disease. What actually is the cause? We didn't know earlier for a long time, and only later on we came to know that it is because of the parasitization of a soil nematode. A small nematode is coming and entering into the plant through the root. They are all entering, and it is called Melodygian incognitia. That pest, this nematode is parasitizing, and this is the Melodygian incognitia that is infecting the roots of tobacco plants. Okay, now we could also make tobacco plants resistant to the attack of Melodygian incognitia by a novel technology by name RNAi. This RNAi is also called gene silencing. They are called by different names like gene silencing or post transcriptional gene silencing, in brief PTGS, or RNA interference or RNAi. What is the technique? The technique was invented in 1990s. This is a mechanism that we came to know only recently. We also are having, that is, eukaryotes are also having an intracellular defense mechanism. That intracellular defense mechanism of eukaryotes is the RNAi. Normally, in our body, there will be no double-stranded RNA produced. But a double-stranded RNA can be produced in 3 occasions. That is, when there is an infection by an RNA, especially reverse transcriptase enzyme containing viruses. So we are having a retrovirus attack or a transposable element or a jumping gene which is trying to change its position, which is very rare, from one side to another side. So during the transposition of that mobile genetic element, there will be the chance of production of a double-stranded RNA. They are called transposons. They replicate by an RNA intermediate. Okay, so we'll be having such conditions in which you will be having a double-stranded RNA produced. Whenever you are having a double-stranded RNA produced, immediately this double-stranded RNA is attacked by a special type of RNAs by name dicer. Dicer is RNAs. It will come and cut that double-stranded RNA. That double-stranded RNA is cut into small pieces. Okay. This is the dicer. So, for example, it will be cutting into small pieces called siRNA or miRNA, small interference RNA or micro RNA. So, this is number one protein that is involved. It is an enzyme, RNase. Second enzyme is RISC protein, RNA interference silencing complex, RISC. This siRNAs will be forming a complex with the RISC to form a RISC complex. That is the technique. This RISC complex will be having only one strand of the siRNA, which is called guide strand. The other strand is removed, that will be destroyed later on. That is called the passenger strand. And this will be having a site called organoid. This organoid acts as the mRNAs. It can cut mRNA which is having a sequence complementary to the guide strand. So whenever you are having an mRNA produced in your cell, immediately the RISC complex will come and will inspect the entire mRNA. That entire mRNA will be inspected with the help of the guide strand. That is why it is called guide strand. So, with the help of the guide strand, it is verifying. And once it is finding that they are all complementary, it will settle and immediately the organoid will come and split or digest that mRNA into pieces. So, mRNA is not allowed to express itself. Though the gene is present and producing an mRNA, the character is never allowed to be expressed. That is why it is called gene silencing. The gene is made silent after transcription. That is, transcription is the process whereby mRNA is produced. That mRNA is cut and destroyed. So, that gene is never allowed to be expressed. That is why this mechanism is called RNA interference. That mRNA is interfered. It is not allowed to express, to produce this character. This technique we have studied. This is the intracellular defense mechanism of eukaryotes. Okay. Once we have studied this technique, we started to apply the same technique to the pathogen that is causing the root node disease. Remember, the root node disease is caused by the nematode, Meloidogen incognitia. which require an enzyme to enter into the root. It has to enter into the root by eating away the cells. So for penetrating into the cells, it requires an enzyme, polygalactosuronase. So it has to produce that enzyme. We have studied that. We took that gene. Okay, we took the gene for the enzyme that enabled the parasite to enter into the cells. And we made two copies of the same gene in such a way that one gene will produce a strand of RNA, which is called the sense strand. The other will be producing the opposite, the complementary strand, which is called the antisense. So this is the sense strand. So one DNA will be forming the sense strand, the other will be producing the antisense. Since sense and antisense strands are complementary, they are not same. They are complementary only. They will be immediately forming a double-stranded RNA. So what we did was, using Agrobacterium tumefaciens, our genetic engineer, we introduced two DNAs into tobacco plant cells. We introduced two genes in such a way that one will be producing sense strand of RNA, the other DNA will be producing the antisense. So when these are all formed, since they are all complementary, immediately they will be forming a double-stranded RNA. And whenever double-stranded RNA is produced, immediately the dicer is coming and activating, and a risk complex is produced. And that risk complex is remaining in the cells of the tobacco plant cells. When such tobacco plant cells are cultivated, what will happen? When the nematode is coming and trying to enter into the root of the plant, they will produce the mRNA for the production of enzyme. See, it is with the help of that enzyme that it is entering into the roots. So it will be producing mRNA. Immediately, the mRNA will be destroyed by the risk complexes that is already made available within the cells by our technique. So that even if the gene is present, mRNA is produced, mRNA will not produce any enzyme. The enzyme production is suppressed, gene is silenced. That expression of the character is suppressed. So no enzyme is produced, and without enzyme, the nematode cannot become a parasite. Thus, the plants are made resistant to the attack of parasites. Is that clear? So that is yet another type of making plant resistant to the attack of pests and parasites. Okay. Now, next to agriculture, human health is important. And we are all trying to produce so many different types of therapeutics, medicines, which are all highly important for our diagnosis and treatment. So, safe and effective therapeutic drugs is one of prime concern of human beings always. And insulin production is a classical example. We started in 1980s and we became successful. Today, there are more than 30 recombinant such therapeutics produced from genetically modified organisms, which are all approved for human use, of which 12 are available in India. They are all presently being marketed in India. The classical example is the genetically engineered insulin. People are suffering from deficiency of insulin or resistance of insulin by the body cells. That condition is called diabetes. Simply we call it as diabetes. Diabetes mellitus. Type 1. It is a autoimmune disease where the patient is completely depending on insulin. Diabetes mellitus, which is depending on insulin. So insulin dependent, it is called insulin dependent diabetes mellitus or type 1. And the type 2 is, as we become old, the cells may become resistant to insulin or the insulin production become reduced. That is called type 2 diabetes. In this, the only remedy is to provide insulin. How? Through injection. We cannot give it through oral because insulin is a protein. It becomes digested. Once it is digested, it reaches on the stomach, it becomes inactive. It loses its ability to act as a hormone or an enzyme. Okay, so it cannot be given orally. So always through intramuscular injection. And what was the source of that insulin till 1980s? We were collecting from butchered animals, from pig or from cattle, and we are all purifying them. See, we cannot survive without them, so we have to get it. And for that, we were using insulin pancreas that are all collected from the animals like pig or bovine insulin. That was creating a problem. What was the problem? Remember, it is an entirely different genus. When that animal's insulin is given to us, it is not of that same efficiency. Number 2, it may cause some immunological responses because we are taking the protein of a different host, which is different from your genetic makeup. So it may activate your immunological systems. So these were the problems when we were all using the insulin taken from other animals. So we were thinking, why not you give that insulin gene to bacterium and make them produce insulin for us? Then there's a problem. Our insulin is produced by a long gene, which is very long, and it is having different parts. We know that our mature insulin is having 2 chains, alpha and beta. It's a heteropolymer. And these alpha and beta chains are interconnected by disulfide bond. But during its production, it is the form of a single very long chain having a middle connector peptide. It is the form of a single long chain with a connector peptide or C peptide. That's how that insulin is produced. And this proinsulin, which is not at all active, is to be cut and edited. During that edition, cutting and editing, that middle piece, C peptide, longest part, it is having 33 amino acid length. This piece is removed and the first and the last part are retained. Okay, so here you will be having a cut. And here. And the middle piece is cut and the first and the last part are retained. Then it becomes a fully mature active insulin. And if we are all to give that insulin gene, only proinsulin can be obtained, not mature insulin. Because in a prokaryote, there is no editing machinery. Only advanced animals are having that editing machinery. So it cannot be edited. We will be getting only proinsulin. and pro-insulin is of no good because it is not functional, it is having no quality of insulin. So, how to get a mature form of insulin was our prime concern. Okay, now Eli Lilly and company in 1983 came out with a technique. We are having the amino acid sequence of alpha chain and that of the beta chain. It was the great man Frederick Sanger who sequenced the insulin and found out what are the amino acids present in alpha chain and beta chain. And also we are having the technique of preparing artificially the DNA. Once you understand the amino acid sequences, what will be the code needed for producing such a type of protein? We already have that technique. So, using these techniques, we understand that alpha chain and beta chain is prepared by two parts of the same genome, which are having specific codes in an order, and we prepared that artificial DNA segment that will be coding for alpha and beta chains separately. So, Eli and Lilly prepared artificial DNA for making alpha chain and beta chains separately. These two artificially prepared DNA, that is genes, were given to two different states of E. coli. They were all cultured and they produced alpha chains and beta chains successively. After purification, these alpha and beta chains were put in a chamber, and when some suitable conditions are given to them, spontaneously disulfide bond was produced between them, and thus it became functional insulin. That was the technique they developed. Eli and Lilly thus became successful in producing the first human insulin, which they call as humulin. Humulin. So, this American company prepared two DNA sequences, which are all corresponding to A and B chains of human insulin, and introduced them to the plasmids of E. coli. So, using E. coli, two separate E. coli varieties or cultures, they produced the alpha chain and beta chain separately, and when these two chains were extracted and combined by creating disulfide bond, they successfully produced human insulin, which is exactly the same as that of our natural insulin. Thus, millions and millions of diabetes patients are now free from anxiety and tension because they are all sufficiently getting insulin. Okay, next use of biotechnology is how to replace a abnormal gene with a normal healthy gene. That replacement of abnormal gene with a normal healthy gene is called gene therapy. We know that we are all suffering, a lot of people are suffering from a number of genetical diseases, which are all because of gene mutations, and there is no remedy. And these gene mutations can be corrected to a certain extent by replacing that mutated gene with healthy ones. That type of treatment is called gene therapy. So, genes are inserted into the person's cell and tissues to treat a particular disease. And the first clinical gene therapy was given in 1940 to a 4-year-old then by name Ashanti De Silva, who was suffering from a disease The disease is called severe combined immunodeficiency, SID. Severe combined immunodeficiency disease. This is an immunodeficiency disease because this patient will be suffering from the absence of an enzyme called adenosine deaminase, ADA. Why there is no ADA? Because the gene for ADA is mutated. The disorder is because of the deletion of the gene for ADA. In some children, ADA deficiency by enzyme replacement therapy can be done. By bone marrow transplantation, it can be treated, in which a functional ADA is given, okay, or the bone marrow is replaced. That is possible, but it is not a permanent cure. The problem of both these approaches that are not completely curative, because the enzyme will be acting only for some time. So again, the patient is to be given that enzyme. Or when bone marrow cells are given, that bone marrow cells will be producing blood cells only for some time. They are replaced by new ones. So these are not advisable, will not be giving you permanent treatment. These are all the techniques that were all used. But we approached a new method. At the National Institute of Health, Dr. French Anderson and his team first successfully made a replacement of defective gene by healthy gene. How? For that, they prepared a recombinant retrovirus vector. That recombinant retrovirus vector was actually a retrovirus which was carrying the single strand DNA for ADA. Okay, thus a functional ADA, complementary DNA, single stranded DNA using a retroviral vector was introduced into the cells of that girl. Which cells? Lymphocytes. So after admitting to the hospital, her blood was taken and lymphocytes were isolated, and that lymphocytes were infested with this retrovirus vector, and this retrovirus vector introduced a complementary DNA for ADA. So ADA cDNA functional was introduced, thus using a retrovirus into the lymphocytes. And after incorporating, these lymphocytes were given back into her body. So these lymphocytes are now having functional ADA gene, and that functional ADA gene will be producing the ADA. ADA is an enzyme that is highly essential for the production of nucleic acids. In absence of the ADA enzyme, nucleic acid multiplication, duplication cannot occur. That is the problem. So the ADA gene was now functioning in her body, started producing the ADA enzyme, and she had normal immunity. But again, there is a problem. Lymphocytes will be living only for a short period. It is not immortal. Lymphocytes will be, our blood cells will be replaced by new sets of cells after some time. So these lymphocytes, treated lymphocytes were also replaced by the new ones, which are again having the same problem. They were all also lacking the ADA gene. So again it is to be repeated. So the drawback was that we had given the gene therapy to the lymphocytes. We had not given it to the bone marrow cells in the embryo. Suppose we were all trying and becoming successful in transferring ADA gene to the bone marrow cells at an early embryo stage itself. When we discover that the baby can have this disease when born, by some technique we are all finding it. Okay, that gene is mutated like that. And if it is done to the embryo at an early embryonal stage to the bone marrow, then that will be a permanent one, a permanent cure. Since we became successful in giving the functional gene to a 4-year-old already formed, already born baby, 4-year-old baby, we have to repeatedly do the gene therapy with it. Suppose we are all successfully doing it to the bone marrow cells and a fetus, then we expect that we will be able to achieve permanent solution. If it is done at an early embryonic stage, it would be a permanent cure. Okay. So we have successfully treated a genetic disease by replacing an old defective gene by a healthy one. Now, we are also using biotechnology in diagnosis techniques. What is our current mechanism? Only when the disease is showing its symptoms, we go for diagnosis. We take our blood or urine and analyze it. Then we identify what is the pathogen, what is the disease, etc. So all these symptoms have established by the time pathogen had become very much grown up in your body. Suppose this pathogen can be traced even when they are all very small in number in our body. They have not started producing any symptom of the disease, but they have just entered and started to increase. Even at that stage, suppose we are all able to identify the existence of a pathogen, identify that pathogen, and if we are treating it, it will be very, very effective. That is what is called molecular diagnosis. Diagnosis at the molecular level. So, even a small amount of pathogen like bacteria or a virus, they are all having nucleic acid. They can be detected by amplification of that nucleic acid by PCR. We have studied that PCR is a technique where we are all amplifying a DNA molecule to millions and millions of times. So even if there is one bacteria or one pathogen in your body, a virus, its DNA, if it is multiplying in a PCR, that shows that there is already a pathogen in your body. So thus we can identify the existence of a pathogen by PCR. Okay, and PCR is now routinely used to detect HIV in suspected AIDS patient. Now, another method is ELISA, enzyme-linked immunosorbent assay. It's a very sensitive antigen-antibody reaction. It can be used either for detecting an antigen that have entered your body or detecting whether your body has started producing antibody. The body has started producing antibody against that particular pathogen. So we can use it either for detection of an antigen or for the detection of antibody. So two-way, dark and DAS method. Okay, we are having these two methods. Here we are using a chromogenic substrate linked to an enzyme in immunosorbent assay. So we are having the antigen-antibody interaction, which is the basic principle of ELISA. By infection, you become sick, and that infection can be detected by the presence of antigens or by detecting whether antibodies are sensitized against this pathogen in the body. And the third method is called autoradiography. In autoradiography, we are detecting whether your genes are normal for a particular character or if the gene is mutated. We know that there are a number of characteristics which are all caused because of gene mutations. So whether we are having a chance of becoming sick because of the gene mutation. Is the gene mutation already present in our body so that in future there's a chance of we becoming sick? We can detect even before the symptoms appear. For that, we are using a technique called autoradiography. It's a procedure where we are using a probe. What is a probe? Probe is an inspecting molecule, a checking molecule. It is a single-stranded DNA or an RNA which is radioactively labeled. You are allowing your gene to be hybridized with this probe molecule. The probe is a single-stranded DNA or RNA for a healthy character, for a healthy gene. That is taken as the probe and is hybridizing with the, this is called DNA probe hybridization. Then it is autoradiographed. Since it is having a radioactive isotope, it will be emitting small amount of radiations. So this radiation will be making a mark on the X-ray film or photographic film which is made on the surface of that DNA pieces that are prepared from your body. And if bands are appearing in that X-ray film, if bands are appearing, that means the isotope is there attached. Isotope is attached means your gene is normal. Nothing to be worried. Your gene is normal and healthy. Bands appear. And if no bands appear, the probes are not attached. Means your gene is different from the probe. Your gene has undergone a deviation, a change. That is mutation. Your gene is mutated. Because the probe is having the sequence which is complementary only to a healthy, normal gene. When band appear, gene is normal. When no bands, no white bands, it indicates that the gene is already mutated. Thus, we can identify whether a particular segment of the genome gene is normal or mutated. Now, we are also using genetic engineering for preparing animals, which are all generally called as transgenic animals. What is a transgenic animal? An animal whose genetical makeup is genetically altered. So, how it is altered? Because it is made to carry a foreign, an extra gene. We have introduced successfully a foreign gene of our desired character to them. Such animals are called transgenic animals. More than 95% of all transgenic animals are mice. Why we are all producing more mice? Because 90% of the genome of man and mice are same. But also we have produced transgenic rabbits, pigs, sheep, cow, fish, etc. also. Why we are all producing such transgenic animals? So that we can study what is the physiology change, what is the change in the development because of the introduction of a new gene. So we can study precisely what is the role of a gene in normal physiology or in the development. Okay. Similarly, how a disease is appearing, a symptom is appearing, what are the symptoms that are causing, how gene mutation can result in appearance of certain symptoms, and thus study of diseases. Deliberately, we have produced such animals called model animals, which are all helping us to understand how we become changed into sick conditions like cancer, cystic fibrosis, rheumatoid arthritis, Alzheimer's, etc. So, transgenic animals are produced just to study, they act as models for human diseases. Okay, so that we can study on them how we become sick and what are the methods to prevent them, how we can make treatment possible, like that. Another one is we want to get this is the production of organism for getting some desired product from them. Okay. So biological products. Transgenic animals that produce useful biological products. So they are made to produce something that is of use to us. For example, we are all successfully producing human alpha-1 antitrypsin, which is used for treatment for emphysema in other organisms. Similarly, we are all trying to make treatment against PKU, cystic fibrosis, etc. First transgenic cow, Rosie. This Rosie was given human lactalbumin and was made to produce human lactalbumin through her milk, which is very rich. So, protein enriched milk that contains 2.4 grams of protein, human lactalbumin per liter was produced. So, that is very ideal for the babies to grow. The milk contained human alpha lactalbumin and was nutritionally a more balanced product. Okay. So, we are all getting products. We produce Rosie, Tracy, alpha-1 antitrypsin sheep. Okay. We are having large number of such organisms which are all genetically modified, produced, which are all giving something which is essential for our health. Then they are also used for testing the new vaccine we have developed. So vaccine safety only after making trials on such field animals and experimental animals will be able to use it in clinical trials in human beings. Okay, so before that we have to try it on such transgenic animals. Also, the chemical safety test, the toxins, how much is toxic, how much concentration will become toxic. All such studies, toxicity and safety testing are done on genetically modified organisms, because we cannot try them on a natural population, natural organism, because this natural organism will be having some resistance. So we won't be getting the correct data. So we have to make them first sensitive to the toxic chemical and then try on them. Then only the studies will be very, very effective. So when we are all thus genetically modifying organisms, there will be a lot of criticism. People will be, and money is most important thing in all these researchers. So multinational companies will be having more interest in such researchers and reaping benefit, and they will be doing it unethically. So there are a number of ethical issues involved in such genetically modified researchers. That's why Government of India had formed GEAC, Genetic Engineering Approval Committee, which is to monitor and validate all GM researchers and safety of introducing genetically modified organisms for public services. People are having a lot of suspicion and apprehensions about the introduction of genetically modified crops for their public use. And also there is a public anger that multinational companies are making biopiracy and unethical methods. What is biopiracy? Pirating of biological resources. See, when you are all using your inherited knowledge or your biodiversity, that should be compensated. That should be only with the authorization of the government or only with the knowledge of the government and people of that part of the world. But many companies are unethically doing this, clandestinely. By secret method, they are transporting, they are taking away our genetical researchers, our traditional knowledges and biodiversity, and after their researchers, they are all producing, marketing the same thing, saying that it is their invention. That is what is called biopiracy. A classical example for biopiracy is regarding basmati rice. See, basmati rice has been grown in India and other neighboring countries for centuries. And we are having a lot of documents, old documents related to the basmati rice. But a company in Texas in 1997 claimed that they have invented a new variety of basmati rice and they got patency from the US Patent and Trademark Office, PTO. Under this, not only they had the right to market, but also all over the world, and people selling basmati rice could be restricted by the patent. That is, they will determine who can only deal with this selling of this basmati rice. So that was a classical example of biopiracy. How they produce that variety of basmati, which they call as Texmati. It is by crossing the Indian farmers' variety. People in India farmers in India were growing it for centuries here. That was crossed with a semi-dwarf variety. And then they produced a variety that they claim as new, but actually it is not at all a new one. Okay, but they secretly took away the seeds of that basmati rice and they collected the rice from Indian farmers and without any authorization, without any documentation and without compensation. That is why the classical example for biopiracy. So without authorization, without compensation, etc., by the countries and people concerned without compensatory payment. And the same thing also made, several attempts are also made in other products like on turmeric and neem. Turmeric has been used in India as an antibiotics. Neem as a neem oil, very effective pesticide. So many companies are trying to market it, saying that it is their invention. So such things are occurring all over the world. Such biopiracy has to be discouraged. Okay. So injustice, inadequate compensation and benefit sharing between developed and developed countries are always there. Developed countries are rich in biotechnology, but are poor in bioresources. Third world countries are poor. Economically, they are poor, but are having rich heritable knowledge, they are very rich in the traditional knowledge and in biodiversity. So, if the developed countries are to make use of it, then they should be given proper compensation. Okay. So, Government of India has promulgated rules in Parliament, and even the second amendment of the Indian patent bills have been passed. So government is always watching such and are bringing about modifications and amendments in terms of emergency provisions and research and development of such genetically modified organisms. Clear? Now, along with this, we'll be discussing a part of the 6th chapter, which is the Human Genome Project. What is this Human Genome Project? Human Genome Project is a mega project. This Human Genome Project involves the study of the entire genome of human beings. Why? It is a mega project. Mainly because it involves a lot of money. We estimated that we will be having a total of 3.3 billion nitrogen bases in the entire genome, and for identifying one nitrogen base, there is a cost involvement of $3 American. So total more than 9 billion. It's a lot of money. 9 billion American dollars will be the expense. And we expected to complete the work in 13 years. It also requires the input of so many technicians, scientists, computer technologists, data analysis, etc., etc. So it is a mega project. And the work started in 1990. Okay. And what are the aims of or goals of this project? We have to identify all the nitrogen bases. How much is the correct number of nitrogen bases that is present in our entire genome? And we estimated that there will be 3.3 billion. Is it correct? What actually is the correct number? And identify how many genes are present. We calculate that there will be 80,000 to 1,25,000 genes in our, that is our calculation. So what actually is the size, what is the total number of genes, and what actually is the size of each gene, how they are all arranged, which chromosome is having maximum number, which is having minimum number, okay, and all such informations are to be stored in a database. And we have to develop the skill in analyzing this data and transfer such technology related informations to industries so that they will be benefited, and ultimately the entire human race will be benefited out of it. And when we start this project, there will be a number of social, legal, and ethical issues, which is commonly called as LC, ethical, legal, social issues, that is to be addressed. These are all the aim of that Human Genome Project. And US Department of Energy coordinates the entire effort, and initially America started in 1990 thinking that they will be able to complete it in 13 years in 2003. And later on, Wellcome Trust of UK became a major partner. Then Japan, France, Germany, China, and other countries also joined it. And thus the project was completed in 2003. And we had a revolutionizing information. We collected a lot of information. Along with that, not only we achieved that goal, but also we achieved some extra goals. What are the two main extra goals? Along with the development we made on the field of technology, see, living things and technology, informatics, information technology was coordinated, and that paved the way for the start of a new branch of biology called bioinformatics. Bioinformatics, a rapid development of a new branch called bioinformatics. Second thing, not only we completed the human genome study, but also we could study the genome of a number of other animals which are called model animals and plants. Thus, we studied bacteria, yeast, a free-living non-pathogenic nematode by name Xenorhabditis elegans, Drosophila, plants like rice and Arabidopsis, etc. They have been also successfully sequenced. And what is the technique? The technique involved two methods. In the beginning, we attempted a technique called expressed sequence tags. That is, we took only mRNAs. Why we took mRNAs? It is easy to sequence that mRNA because it is single-stranded short pieces. And we collected them and studied their sequences. But only then we came to know that it will not be giving you completely accurate knowledge on our genome because only 2% of our DNA will be coding for mRNA. So even if we completely study all the mRNAs, we will get information only on 2% of our genome. 98% of our genome are not coding for any protein. So this technique we changed to this technique was not sufficient for our information, and we developed another technique called sequence annotation. In this second technique called sequence annotation, whether they are expressing, that is producing mRNA or not, without any such regard, we are taking the entire genome and with the help of computers, we are all studying the genome. And for that, we had two steps. Okay. What are the first step? In the step 1, we are all identifying the sequencing of the genome. That itself is done in 2 steps, A and B. In the first step, we are all sequencing short DNA fragments that were sequenced, and these DNA fragments were then aligned. Then these pieces were aligned properly, and thus the entire sequence of the DNA was known. These two were done with the help of computer technology. They are all computer assisted. Okay. Here, in step 1, we will be taking the DNA. Okay. From a particular chromosome, we started. We started from the smallest chromosome. Okay, 22nd pair, which is the smallest, and proceeded gradually towards the first chromosome, which is the largest, because taking smaller one is easy. And as we progress, as our technology become more and more familiar and foolproof, we can do the larger pieces, larger chromosomes with much ease. So that was our approach. So DNA was taken, then it is cut with restriction enzyme, restriction enzyme fragmentation. Then they were all resolved in an agarose gel electrophoresis. They were all sequenced. They were all separated, resolved in agarose gel electrophoresis. Then we gave it to the artificial vectors like bark, bacterial artificial chromosome, yeast artificial chromosome. Later on, when we invented PCR, we abandoned this cloning and instead we used PCR. So that very conveniently you can get large number of copies of our DNA segments. So after that, they were treated with sodium hydroxide alkali treatment. So double stranded DNA will be separated, and sometimes before sodium hydroxide treatment, they were transferred to nylon paper by southern blotting. Southern blotting is a technique for transferring the DNA pieces from the resolved agarose gel electrophoresis to nylon paper and then treat with sodium hydroxide so that they become single stranded DNA, and then that single stranded DNA was given to automated sequencing machines. That machine worked on the principle developed by Frederick Sanger, who is the first man to develop a technique for sequencing both a protein as early as 1955 as well as for sequencing nucleic acid in 85 he discovered. So this great man discovered the technique of sequencing a nucleic acid, and an automatic machine was discovered later on which will be automatically telling you what are the nitrate bases present in that single stranded DNA you have fed that machine. That can be stored. Remember, suppose we were all writing all the nitrate bases of the entire genome project. I told you we will be having 3.3 10 raised to 9 nitrate bases. That was our estimate. That will be requiring a total of 33,000 books or 3,300 books. 3,300 books. Each book will be having 1,000 pages. Each page will be having 1,000 informations. Okay, this much of data will be generated, and that is why I told you it is a very mega project. Now this data were fed to the computer that will be making a database. Now what we got is the sequences of different pieces which are all separate based upon their sizes, not on their position. Then they are to be aligned properly. That is the second step. This alignment is based upon some overlapping sequences. It is based upon some overlapping sequences. We deliberately produced overlapping sequences by digesting the same DNA with two different restriction enzymes and sequencing from both ends of each pieces, thus making it foolproof. Now the computer will tell what pieces are having the same overlapping sequences, and it will automatically bring these elements based upon these overlapping sequences. If the two segments are having the same overlapping segment sequence, they will be brought together, and thus small pieces are joined together and will be forming long chain. Thus the entire sequence is studied. Then comes the second part. Okay, what is the second part? Second part is not based upon the, this is the first part, not based upon the computer application, but it is based upon two. It is sequence annotation. We are making physical map and genetical map. We are all marking which is the position of a particular gene, what is the size of that gene, what is the position of that gene, and what are the genes that are present in a chromosome, etc., etc. That is what is called physical mapping or genetical mapping. That is not assisted by computer, but it is based upon some characters like DNA, restriction enzyme, polymorphism, and based upon some microsatellite DNAs. So it is based upon these two properties. Microsatellite and restriction endonuclease recognition polymorphism. So based upon these properties, will be mapping correctly what is the position of a particular gene, what is the size, where it is situated, etc. Thus, we completed the Human Genome Project. And through that, we came to know that our genome is having a number of features. The correct number of genome, nitrogen base sequence in our genome is 3164.7 million base pairs, not 3.3 billion, only 3.1647 billion. The size of an average gene is 3000, but will be having some genes which are as large as having 2.4 million bases. We actually have about 30,000 genes. We didn't have the estimated 80,000 to 1,40,000 genes. And remember that 99.9% of the nucleotide bases are same for all people. That is, we differ only in 0.1%. And the functions are only known for 50% of discovered genes. Only 2% of the genome code for protein. What is the role of others? We have to study. And large sequences, repeated sequences, which are coming repeatedly, will be forming very large portion in our human genome. And these repeated sequences are stretches of DNA segments that are repeated many times, sometimes hundreds to thousands of times. They have no role in coding. That is, they are not expressed, but they may shed some light on the chromosome structure, dynamics, and evolution. The largest chromosome, chromosome number 1, has the largest number of genes, 2,968, and Y is the chromosome which is having the smallest number. And scientists also identified that in 1.4 million locations where single base DNA differs, SNPs, we call it as SNPs. It is difference is only in one nitrogen base in 1.4 million locations, positions. Clear? Thus, we successfully completed the mapping of the first chromosome in 2006. Though we expected to complete in 2003, by the time we mapped the first chromosome, it was 2006. Even though we were a bit late, we successfully completed the Human Genome Project that revolutionized our idea on our genes and also concept of our genetical studies. Second part is DNA fingerprinting that was developed by Alex Jeffrey and his colleagues. In human DNA or human genome project, we came to know that 99.9% of our DNA are similar. Only 0.1% is different. And only 2% of the DNA code for character, that is 98% are not coding. And that part is mainly made up of tandem repeats. Same repeating sequences that comes in thousands and thousands of times. They are not coding for any protein. And as they are not coding for any protein, any change that occur in that part will not be bringing any change in the character of an organism. A mutation that is occurring in a expressing part will definitely bring about a change in the character, which is very often deleterious or harmful, and natural elimination will eliminate them. Natural selection will eliminate them in due course of evolution. Within a few generations, such mutation will be eliminated from the population. Whereas mutations that are occurring in the non-coding part, mutation that is occurring in that part which is not at all responsible for a particular visible character will be remaining in the population for thousands and thousands of generations without any change. So, you are having more change in the non-coding part. So, it is the non-coding part that you have to look into to distinguish one DNA with another DNA. So, the difference, the uniqueness of my DNA lies in my non-coding part, because that part is containing maximum difference. Difference at the molecular level is called DNA polymorphism. DNA polymorphism more occur in the non-coding part. It is the non-coding part which is made up of tandem repeats. So, when we perform a autoradiography with this tandem repeat, which is different from one individual to another, you will get the unique features of the human DNA. Thereby, you can compare DNA of different persons. DNA of a man will be unique in the non-coding part in the nature of its tandem repeat. This was the idea developed by Alex Jeffrey. And this technique he called it as DNA fingerprinting, just like our fingerprints. Just like our fingerprints, which are all unique for our, that will be never changing. Our characteristics of this part of the DNA in the non-coding tandem repeats will be never repeated in anybody else in this world. It is unique for me. So that can be used for identification of my genome. So how you will be getting that? It is simple. You perform an autoradiography of your particular DNA of an individual. Remember, all cells of same individual will be having the same DNA polymorphism. Whether it is a WBC or a saliva cell or sperm or a hair, all these cells are having the same polymorphism of DNA. So, any cell is sufficient to identify. DNA polymorphism will be different for different chromosomes. It is not same for all the chromosomes. From chromosome to chromosome, the polymorphism will be differing, but it is unique for all cells. Same chromosome of all cells of that individual. So any cell is sufficient. So you have to take a cell with nucleus. Then just like you are all doing your autoradiography, you take that cell, isolate that DNA, cut it with restriction enzymes, resolve it, and after resolving, you can have PCR. So even if you are getting only a single piece, you give it to the PCR, so you will be getting thousands and thousands of copies, or you can give that to the PCR after your transfer by southern blotting to a nylon paper. Okay, so you will be getting large number of copies, millions and millions of copies of the DNA segments. Okay, then treat it with sodium hydroxide. So the double stranded DNA becomes separated and becomes single stranded. Then you use a probe. What is the probe here? Alex Jeffrey used a mini satellite DNA. What is a mini satellite? The DNA can be isolated by density gradient centrifugation method, and the DNA that is coming out, denser one, is called the core DNA, which is very rich in CG pairs, and the mini satellite or the mini peak, small peaks that are coming out will be the lighter DNA, which is rich in AT pair. So that second peak, minor peak, which is called satellite DNA, are coming out. This satellite DNA can be classified as, based upon their size, mini satellite and micro satellites. Mini satellites are DNAs which will be having a size of about 0.1 to 20 kilobases. Alex Jeffrey used a mini satellite that shows maximum polymorphism, which he called as VNTR, variable number tandem repeat. He used a VNTR, a satellite DNA that shows high degree of polymorphism. That was his probe. He made them radioactive and used it as a probe for hybridizing with that single stranded DNA you have already prepared. And when X-ray was placed on that and covered and incubated, that is, we are having autoradiography done. In that X-ray film, you will be having bands of special type or special nature, which is unique. Okay, that markings on the X-ray film is called the DNA fingerprint. Your fingerprint is unique for your DNA. It will be never the same for anybody else. Even for identical twins, there will be some differences. Even you are having, say, for example, identical twins, there will be some differences. Okay, so this is the technique used here. Isolation of the DNA, digestion with restricted enzyme, separation with heterophorsis, blotting, hybridization using labeled, radioactive labeled VNTR probe. Nowadays, we are not using VNTR, but instead you are using highly specialized, artificial prepared probes, which are all unique for each research laboratory. They are all invented by that research laboratory, patented by that research laboratory. So such probes are used here. Okay, and that is capable of even detecting even a difference in one nitrogen base. So sensitive. Probes are nowadays used. And this technique is nowadays, see, for chromosome 7, paternal, maternal. So the baby will be having either of these two inherited. Similarly, either of these two from the maternal or paternal. Okay, for each chromosome, the binding pattern will be different because I told you polymorphism is different for different chromosomes. So DNA from individual A, this is DNA from individual B. So you can see that the B is more similar with that of the C. Number of short tandem repeats, okay, their size, etc. So from this, we can identify that B is more similar to that of C. Thus, this technique became very important tool in deciding the disputed parentage problems. Even courts are accepting this technique as a scientific tool in clearing disputed parentages. It is also helping in forensic studies, in identifying the criminals. We are also using them. So this has been developed as a scientific method for settling disputed parentage as well as in forensic studies. Okay, so that is a new branch that is developed because of the progress we made in biotechnology. So with these words, I come to the conclusion of the discussion of this chapter, biotechnology. I hope we could cover almost all the topics coming under biotechnology, and next time we will meet again with a new chapter. Until then, goodbye and thank you all. Thank you very much.